Hey everyone, Dr. D here, and in this video we are going to be covering Chapter 15, the final chapter of 1406 of the semester. So let's go ahead and get started. Chapter 15, what we're looking at is genes and how they work. So I've been telling you throughout the semester that genes are the genetic information that codes for a particular protein. And this is called gene expression, how the DNA, a gene, eventually codes for a protein. Remember what I've been saying, DNA. The reason you have DNA, the reason you have chromosomes is because that contains the genetic information, the genes. And each gene is, this is a general statement, but for the most part, each gene is the information for a particular protein. Okay, so for example, the insulin gene would give you the information you need to build the insulin protein, which would go off and regulate blood sugar. Now, what you need to understand is proteins are made from the information on your genes, but not directly. First, your genetic information, your gene information is copied into complementary RNA. Okay, that's step one of gene expression. And then the RNA information, which is essentially that gene's information, that RNA information is translated to protein. So there's two main stages here, DNA to RNA, RNA to protein. The first stage, DNA to RNA, is called transcription. Okay, transcription, DNA to RNA. Again, what are you doing during transcription? You are copying the gene information to RNA information. And the RNA in this case is called mRNA for messenger RNA. Okay, transcription. Then, in the next stage, you take that mRNA, messenger RNA, and the ribosomes will translate that message to protein. That's called translation. Translation is where the mRNA message is translated to an actual protein. And together, this is the central dogma of molecular biology, that the information flows from a gene on your DNA to mRNA to protein. Okay, so transcription again. Transcription is the process that produces the messenger RNA or mRNA. And then translation is the synthesis of a polypeptide. Remember, that means a protein from that mRNA message. Ribosomes are responsible for translation. So here it is. It doesn't matter if we're talking about prokaryotes. It doesn't matter if we're talking about eukaryotes. They both have genes that they carry on their chromosomes. And that those genes are composed of nucleotides. Remember, G's, A's, T's, C's. And each gene is the genetic information for an mRNA. Okay, and then that mRNA is translated to a particular protein. Remember, the first step from DNA to RNA is called transcription, and then from RNA to protein is called translation. Now take a look at this. This is how gene expression works in a prokaryote on the left and a eukaryotic cell on the right. There's key differences between gene expression in a prokaryote and a eukaryote. Okay, there's key differences here, so we're going to talk about those right now. All right, so in a prokaryote, you've got your chromosome. Remember, you have one circular chromosome. On that chromosome, you have all of your genes, right? Thousands of genes. Okay, so let's say this stretch of DNA is a particular gene. It codes for a particular protein. So you would copy one of those two strands of DNA called the template strand. You copy one strand into mRNA, complementary mRNA. That mRNA is then read by the ribosomes and translated into protein. Remember, making the mRNA was called transcription, and then reading the mRNA message to make a polypeptide, a protein, is called translation. And what you need to know about prokaryotes is that transcription and translation both occur at the same time at the same place. What does that mean? They both occur at the same time, which means transcription starts. But what you need to realize is in a prokaryote, before transcription is even finished, translation can start. So they're happening almost concurrently, right? And they're also happening in the same place. What is that place? Well, it's happening in the cytoplasm, the fluid inside of the cells. Okay, now compare that to a eukaryotic cell, which has a nucleus. Remember, this purple structure is the nucleus. In a eukaryotic cell, you've got your, chromosomes, multiple chromosomes that are linear, like a human cell would have 46 chromosomes, and each chromosome has hundreds or thousands of genes on it. Okay, and a gene is just, like I said, the information for a particular protein. So here we have part of a chromosome called a gene. It's in the nucleus, isn't it? Remember, our chromosomes are inside of the nucleus. So where does transcription have to happen? That's right, in the nucleus. Transcription, so you take the template strand of this DNA, you copy that information to complementary RNA. This now, look at this. Is it called mRNA? No, if you look closely, it's called pre-mRNA. You know why? Because the mRNA is not ready for translation. You actually have to do an additional 3 steps in order to process the pre-mRNA. Look, the pre-mRNA is not ready for translation. It needs to undergo RNA processing, which is 3 steps, which we're going to talk about in a little while. That produces the mature mRNA, which has been modified. The mature mRNA is then able to leave the nucleus, go out into the cytoplasm where it meets up with the ribosome. Remember, the ribosome is the organelle that undergoes translation. Its job is to do translation. So the ribosome attaches to the pre-mRNA at the 5' end over here, and then it would copy that information. Actually, not copy, but translate that information into an amino acid message, forming a polypeptide, which is a protein. So again, to summarize, where does transcription happen? In the eukaryote, in the nucleus. Where does translation happen in a eukaryote? In the cytoplasm. And do they happen at the same time? No, you have to complete transcription and you have to process your pre-mRNA into mRNA beforehand, and only then, after exporting that pre-mRNA, can you do translation. So to review, prokaryotes, transcription and translation happen at the same time and at the same place. In eukaryotes, transcription happens first, then pre-mRNA processing to mRNA happens, and then and that happens in the nucleus, and only then is the pre-mRNA transported out into the cytoplasm for translation. So transcription and translation do not happen at the same place, nor do they happen at the same time in eukaryotes. Okay. So let's get to the important slides here. So the genetic code. How does your gene code for proteins? Okay, this is called the genetic code, how your genes code for proteins. Remember, your genetic information is composed of 3 letters, right? You got A's, G's, C's, and T's, right? But how many amino acids were there? Were there only 4 amino acids? No. There are 20 amino acids, right? 20 amino acids, but only 4 nucleotides, A's, G's, C's, and T's. So if the DNA codes for proteins, how does it code for proteins? There are 20 different amino acids, but only 4 different, nucleotides or nitrogenous bases. Well, what Francis Crick and Sidney Brenner figured out was that the genetic code works in blocks of 3. There are blocks of 3 DNA nucleotides, and they called this the codon. Codons are triplet code, 3 nucleotides in a row. So AAA is a codon, GGG is a codon, GAC is a codon, CAT is a codon. Any 3 letters composed of C's, A's, G's, and T's That's a codon. And what they figured out was that each codon, each block of 3 nucleotides in a row, codes for a particular amino acid. Okay, so if we're talking about, I don't know, the amino acid methionine, which is one of the 20 amino acids, its code is ATG, right? So if you have ATG, that codes for the amino acid methionine, and there you go. Okay, so you can see here there are certain codons besides codons that code for amino acids. There are the stop codons. What is a stop codon? These are the codons that code for stop translation, stop making a protein. That's UAA, UGA, or UAG. On the DNA, that would be TAA, TGA, and TAG. And there's also a start codon, AUG, or on the DNA would be called ATG. This is the start codon. So let me show you the whole codon table. You see here, this is the entire codon table. What does that mean? It means every possible 3-letter word that you could make with A's, G's, C's, and T's. But here on this table, they're showing you U's instead of T's because they're talking about the RNA. Either way, it doesn't matter if you're talking about U's or T's. They mean the same thing. So here you've got U's, A's, C's, and G's, which is every 3-letter word you could possibly make in the genetic language. Okay, and also you can see here, here's the first letter of the codon. Then you've got the second letter of the codon, the third letter of the codon. So the first amino acid, or the first codon, is U, U, U. What amino acid does that code for? Phenylalanine. The next codon is U, U, C. What does that code for? Also phenylalanine. UUA codes for leucine. UUG codes for leucine. So you see what you could do? You could find any codon like CAG and figure out what amino acid that codes for. CAG codes for glycine. ACG codes for threonine. GGC codes for glycine. Remember, there's 20 different amino acids, but there's actually 64 of these codons. If you counted, look, UUU, UUC, UUA, UUG, that's 4 codons. But how many codons are in this whole codon table, which is every possible 3-letter word? 64. So there are 64 codons, but how many different amino acids? 20. So you know what that means? Some of these have to be redundant or degenerate. Does that make sense? Like, look, CCU, CCC, CCA, CCG, they all code for the same exact thing, proline. UCU, UCC, UCA, UCG, they all code for serine. So you know what that means? The genetic code is degenerate or redundant. Do you know what that means? Degenerate or redundant. More than one codon may specify a particular amino acid. Okay, so you see that? You have more than one codon specifying one amino acid. However, the genetic code is not ambiguous. You know what that means? Not ambiguous. CCA codes for proline, but does it code for any other amino acid? No. CCA only codes for proline. So the genetic code is not ambiguous. No codon specifies more than one amino acid. So what you need to know for exam reasons is the genetic code is degenerate or redundant because you can have more than one codon that codes for a particular amino acid, but the genetic code is not ambiguous, which means when that codon specifies one thing, it doesn't specify anything else, any other amino acids besides that very specific one. Okay, so this is the genetic code here. These are the codons and the amino acids that they code for. And do you remember what I showed you here? There are 3 stop codons. This tells these codons on the mRNA would tell the ribosome you're done making a protein. That's UAA, UGA, UAG. So let's take a look at those. You see here, UAA, UAG, UGA. These are all stop codons. Do these code for an amino acid? No. These code for nothing. These tell the ribosome that the protein is finished. Okay, those are called the stop codons. So 3 of the 64 nucleotides, they don't code for any amino acid. They just tell the They tell the ribosome that the protein is complete. But what was this other thing? The start codon. What is that? The start codon, AUG, or on the DNA it would be ATG. What is the start codon? The start codon, this one, AUG, is the start codon. It tells the ribosome to start making a protein, okay? And does it code for an amino acid? This one does code for an amino acid. The amino acid is methionine, or M, okay? So just be aware of that. There's a start codon. That start codon codes for start, and it codes for the amino acid methionine. There are 3 stop codons, and that does not code for an amino acid, but it does code for stop translation. Now, the rest of these codons all code for different amino acids. Okay, so again, the genetic code is degenerate or redundant, but it is not ambiguous. Okay, at this point, let me take a quick break, maybe grab a drink. I'll be right back to explain further. Welcome back from break time. So where we left off, I had told you about the codon table, and what we're going to get to now, we're going to keep going. We saw that you have double-strand DNA, and that DNA houses your genes, which is the genetic information for a protein. Now, what you need to understand is that only one of the 2 strands of DNA is copied into mRNA, and that is called the template strand. So you make a complementary copy of the template stranded DNA. A's get copied to U's, C's to G's. All right, so you form your mRNA, which has a 5' end and a 3' end. And the codons can be found on the mRNA, you see? You have UGG, that's a codon, UUU, that's a codon, GGC, that's a codon, and the different codons code for the different amino acids by the ribosome during translation. Okay, that's the protein right there. So remember, all you have to do is look at your codon table here, and then UGG is the codon that codes for tryptophan, UUU codes for phenylalanine, GGC for glycine, UCA for serine, and so on and so forth. So that's what your DNA basically looks like. Your chromosomes are one giant DNA molecule. On that DNA molecule, different segments of it are the genes. So you have gene 3, gene 2, gene 1, and it's kind of how it works. Now, a really interesting thing about the genetic code is that it is practically universal. What does that mean? That means that the way that the codons are deciphered in one organism is the same, usually identical, in all organisms. So here you have what's called a transgenic pig, and here is its wild type. Wild type means normal type, normal genetic type sibling. All right, this is a normal pig, wild type pig. This is a transgenic pig. The transgenic pig has an extra protein, and this protein is from jellyfish, so it's yellow fluorescent protein. Okay, researchers inserted yellow fluorescent protein gene into the pig so that its soft tissues and its cartilage produce the yellow fluorescent protein. Now, if you were to turn off the lights and put a black light on, this pig would glow. It would be fluorescence, right? The yellow fluorescent protein would fluoresce, and its snout and its toenails would actually glow, and perhaps its ears as well, because there's cartilage in the ears. So its wild type sibling doesn't have that gene, that extra gene. That's why it looks normal. Now, what does this have to do with the code being practically universal? Well, when they took the jellyfish gene, those codons were found in the jellyfish. When they put that gene into the pig was able to decipher the code the same way as the jellyfish does and produce the correct protein from it. So you see what it's getting at? You see this code and how each code codes for a different amino acid? Well, that code is practically universal. You see ACA codes for threonine, GCA codes for alanine. Well, this codon table is relevant for almost all life on Earth. So the genetic code is practically universal. Hence why you can take a pig and insert it with a jellyfish gene and it produces jellyfish protein. Fun fact, do you know how human insulin is made for diabetics? Uh, they've taken the human insulin gene and they've transfected E. coli or other prokaryotes with it, and the E. coli grow up in these big vats, and they're constantly producing insulin. And then what researchers do, they spin down that E. coli, they break open those cells, they purify that insulin protein, bottle it up, and sell it to treat people. So that would be an example of a human gene being expressed in transgenic E. coli. Very interesting, because E. coli have no business producing insulin, but we use them to produce tons of insulin for diabetics. Interesting stuff. So moving on, I'll come back. Oh, here. Let's go here. So how does transcription actually work? Now that we know what the codon table is like and what gene expression is transcription and translation. How does transcription work? Well, this is an overview of transcription, right? RNA synthesis. Here's a gene. Okay, this double strand is a gene. It's part of a chromosome. Now, what you need to realize is you have to copy this gene, right? Or at least you're going to copy the template strand of this gene. It looks like the bottom strand is the template strand. In this example, the bottom strand is the template strand. So look, this enzyme, if here it's in this kind of opaque white, this enzyme is called RNA polymerase. RNA polymerase attaches to the gene in front of the gene. And the place it attaches to, which is in front of the gene itself is in dark blue here. Okay. Notice where the RNA polymerase attaches right in front of the gene. It's called upstream of the gene. It's attaching to a segment of DNA called the promoter. Okay, so RNA polymerase attaches to the promoter, which is the segment of DNA upstream of the gene. RNA polymerase then has kind of helicase-like abilities. It can unwind the DNA, so basically separate the two strands from one another. And then look what RNA polymerase does. It starts copying the template strand of DNA, the bottom strand of DNA, and copying it into an RNA transcript, copying it into mRNA. And then what happens? Elongation. The RNA polymerase proceeds across the gene, and it's synthesizing the RNA transcript in a 5' to 3' direction until it's finished. The RNA polymerase will proceed until it's finished, and then it will leave. It will pop off the gene, leaving behind a complete RNA transcript. Okay, so this is the process of transcription. And you could see here, this is the RNA polymerase doing its job. We found it in the middle of doing its job during elongation. You see it's unwound the two strands of DNA. It is making a template, it is making a complement of the template strand. You see that? In a 5' to 3' direction. And here you can see it in a little bit more detail. This would be the RNA polymerase here. The RNA polymerase attaches to the promoter. It then separates the two strands of DNA and then proceeds on, okay, to copy the template strand. Okay, and how does it know to finish? How does it know when it's done? How does it know when transcription's done and when to terminate? There's usually sequences on the DNA that tell it to stop, tell the polymerase to stop. You don't need to really know this, but what I'm trying to get at is there are DNA sequences here that tell the RNA polymerase to stop and to finish transcription so that it only copied the information for one particular gene. Now what's really interesting is in prokaryotes, you are able to copy not just one gene into mRNA, but prokaryotes sometimes allow you to, there are certain groups of genes called operon, and those are groups of genes that are functionally related, and they're like kind of concatenarized. They're all right next to each other. So what ends up happening is you have one promoter, but you'd have a number of genes right next to one another, and you're going to copy all that genetic information into one really long mRNA, and that really long mRNA is the information 3, 4, 5 different proteins sometimes. Okay, so you don't need to know the fine details about that, but just realize that in prokaryotes you can have these genetic structures called operon, which is essentially groupings of functionally related genes under the control of a single promoter. So what you could have is, for example, if the bacteria wants to digest lactose, it could have a number of lactose genes in a row that code for all these different proteins required for breaking down lactose. That would be called the lac operon, for example. All you need to know is that eukaryotes do not have operon. Eukaryotes, you have one promoter for one gene, okay? But in prokaryotes, you can have operon, which means one promoter, several genes. And that's essentially what I'm trying to get at. Okay. This is just showing you in a prokaryote how you have the DNA. The green dot is the RNA polymerase copying the DNA into complementary mRNA. And then these little circles, these little yellow circles, are the ribosomes quickly attaching to that mRNA to make the polypeptide, the purple polypeptide. So remember, in prokaryotes, transcription and translation happen at the same time at the same place. Okay, in eukaryotes, how is eukaryotic transcription different? Well, eukaryotes, you have very similar enzymology, but you need, in order to start transcription, you need all these other proteins to attach to the promoter first. Do you remember in prokaryotes? Look at this. This is prokaryotic transcription initiation. The RNA polymerase, what does it do? It attaches directly to the promoter, which has these sequences, and then it starts transcription, right? Well, in eukaryotes, the main difference is, if this is RNA polymerase, first a bunch of other proteins have to attach to the promoter, and that recruits RNA polymerase to the promoter. Okay, so these little proteins that have to attach first are called transcription factors. So what you need to know is that eukaryotes require transcription factors to attach to the promoter before RNA polymerase can attach. However, prokaryotes do not need transcription factors. Okay. And do you remember what else prokaryotes need? Do you remember what else prokaryotes need? I'm sorry, eukaryotes. Do you remember what else eukaryotes need? They need to do mRNA processing, right? Let me show you what I'm talking about real quick. Let's go back to this slide here. Look at this slide here. Remember, in prokaryotes, transcription happened, and the mRNA was already mature. The mRNA was ready for translation. You see that? Great. But look at eukaryotes. Remember, you have the gene, you do transcription, you have the mRNA. Is that mRNA ready for translation? No. The mRNA is called pre-mRNA. It needs to be processed by RNA processing, which means you have to modify the pre-mRNA, right? And you can see two of the modifications right there, the yellow bits. And only then is the mRNA, mature mRNA, ready to leave the nucleus and meet up with the ribosome. Okay, so what are the steps involved in RNA processing? There are 3 steps involved with RNA processing, and only eukaryotes need to do these 3 steps. Okay, let's talk about those 3 steps. Let's see. Okay, these are the 3 modifications of RNA processing, the 3 steps I'm talking about. First, in eukaryotes need to add a 5' cap to the 5' end of the mRNA, the pre-mRNA. Why? Because this will protect it from degradation. Okay, and it is also involved in initiating translation, so it helps with initiating translation. Then the 3' end of the mRNA needs a poly A tail, so a bunch of adenines need to be linked on in a row, a bunch of A's. You know how you have A's, G's, C's, and T's? Well, you got to have a bunch of A's, a bunch of adenines added to the 3' end. Why? Because this protects the mRNA from degradation. What you need to understand is mRNA is susceptible to degradation. It's very susceptible to breaking down spontaneously, right? And then you have the 3rd modification, which is removal of non-coding sequences called introns. Okay, this is a tricky one, but I'll explain it. All right, so we've got 3 modifications: 5' cap, 3' poly A tail, and removal of non-coding sequences called the introns. So let's take a look at the first two. If this is your mRNA in yellow, the yellow is your pre-mRNA. Okay, the 5' end. This is the 5' end. It gets a cap. You see, this is the cap. And you don't need to know a ton about this, but I'm going to go into some detail for you just so you know. This cap consists of guanine, right? G, guanine triphosphate, 3 phosphates, but it's not just guanine, it's methylguanine. Why? Because this guanine has a methyl group here. Okay. So what happens during step 1, addition of a 5' cap? An enzyme comes along, takes methylguanine triphosphate, and it adds it to the 5' end of the mRNA, but it adds it the wrong way around. What do I mean by that? The 5' end with the phosphate groups of the methylguanine is going to be attached to the 5' phosphate on the mRNA. Isn't that the wrong way around? You're adding the 5' end of the methylguanine to the 5' end on the mRNA. So basically, you've taken a G with a methyl group and you've stuck it on the mRNA the wrong way around. Why would you do that? Again, it increases the durability of mRNA, prevents degradation, and it helps the ribosome to find the mRNA and where the 5' end of the mRNA where transcription begins. So it helps with the translation process as well. So that's why we're doing it. Again, you don't need to know it in that level of detail, but that's what's going on. You need to know there's a 5' cap, and that's part of the RNA processing. On the 3' end, there's usually a signal like AAU, AAA on the mRNA, and that triggers this other protein called polyadenylase. You don't need to know that. This enzyme comes along, and post-transcriptionally, that means after transcription's over, this enzyme comes along and adds a bunch of A's, a poly A tail to the 3' end of the mRNA. So you're adding a bunch of A's, and by a bunch I mean a bunch, like 100, 200, sometimes 300 A's to the end of the mRNA. And again, why are you doing that? Because it helps to lengthen the lifespan of the mRNA and help against degradation, like quick degradation. Okay, now that's the first two modifications: 5' cap, 3' poly A tail, and then the last modification is called removal of non-coding sequences or introns. So before I get to that, I may actually have to explain that at the board. So let me head over to the board to explain that now.
All right, so here's something you need to understand about eukaryotic genes. Let's say this is a gene. This is the portion of a chromosome that codes for a particular protein. So for example, if this was the insulin gene, it would code for the insulin protein. Or if this was the hemoglobin gene, it would code for the hemoglobin protein. So what you need to understand is that in eukaryotes, and not prokaryotes, in eukaryotes there are parts of the gene that actually code for protein, and then you have parts of the gene, which I drew here in red, that don't. They're filler. They used to call it junk DNA, but now we know it has other functions besides coding for protein, but it's not parts of the gene that actually code for protein. So the parts that do code for protein are called exons. So this would be exon, EXON1. This one would be the next exon, EXON2. And then you have exon 3. Exon 1, 2, and 3. So this gene in particular has 3 parts to it that code for this particular gene. But then you also have the other filler parts, and the fillers are called introns. So this would be called intron, I-N-T-R, intron, intron 1, intron 2, and intron 3. Intron 1, 2, and 3. So why do we care? Again, these introns, these filler, This filler information in the middle of the gene is not present in prokaryotes. It's only found in eukaryotes. In a prokaryote, the entire gene is like one big exon because everything codes for a protein. In a eukaryote, you have introns. Here's the problem. When the RNA polymerase attaches to the DNA and copies the DNA into mRNA, right? Remember mRNA? What are you copying? When you copy the DNA into mRNA, that pen does not want to stay, you copy all of it, don't you? You copy all of that gene from beginning to end, don't you? Which means not only did you copy the exon information, you also copied the filler information, the intron information. Hey, no, Tig wants in. Come on in, Tig. Yeah, come on in. We're making a video. Now he's sitting outside yelling. Come on in. Yeah, come on in. Okay, or just stay outside and yell. He does that. So anyway, this in a eukaryote, this would be called what? Is it mRNA? No, remember it's called pre-mRNA. Pre-mRNA. So the pre-mRNA, if you recall, this would be the 5' end of the pre-mRNA. This here would be the 3' end of the pre-mRNA. The 5' end gets a cap, right? Cap. A 5' cap. The 3' end gets a poly A tail, right? You get a bunch of A's added on. Remember that? And now for the third and final modification on your pre-mRNA. You see that red information, the intron information? Remember I said that information is really not useful for the ribosome to make a protein. It's not useful information. So we actually have to cut that information. Now imagine if I had scissors, molecular scissors, and I could just cut out the red parts, the intron parts. Because remember, the blue part is exon 1, exon 2, exon 3, right? But you have the red parts, which are intron 1. Oops. I'll just call it I1, I2, and I3. We have to remove the introns. This is called splicing out the introns. A special enzyme called the spliceosome comes along and it literally removes, it splices out, excises the introns. Okay, you need to know that the introns are excised by the spliceosome. The spliceosome is the enzyme that will splice out or excise the intron. So what are you left with? You're going to be left with, if you cut out all the introns, you're going to be left with a shorter, actually even shorter, a shorter mRNA. This short mRNA is called your mRNA, mature mRNA. Here's the 5' with a cap. Here is the 3' with a poly A tail. And it's a much shorter mature mRNA because we've cut the intron information out. Okay, so those 3 modifications, you don't need to know in great detail, but you need to know that in eukaryotes, you have intron information, and when the mRNA is produced, that mRNA is quite long because it includes that intron information. So the 5' end of the mRNA needs a cap. That's called the 5' cap. The 3' end gets a poly A tail, and the third and final step, excision of the introns, right? Removal of the introns by the spliceosome. In the next step, I'll show you what the spliceosome looks like.
All right, so as you can see, eukaryotic genes contain exons and intron information. The exons are valuable for producing a protein. The introns are not. So when the pre-mRNA is made, you see here the purple denotes the intron information. That intron information needs to be spliced out, resulting in a much shorter mature mRNA which only contains exon information. The intron information has been removed. And I told you I would tell you how the introns are removed. The enzyme responsible for removing the introns is called the spliceosome. Look, here's a spliceosome protein. Actually, the spliceosome is part protein, part RNA. You don't need to know that, but just to be specific, spliceosome attaches here in front of, for example, exon 1 and before intron 1, and it attaches here at the end of it, the intron, and before exon 2. So spliceosome proteins attach here, spliceosome attach here, and then look what happens. The spliceosome is able to loop the intron, and look what the structure is called. This loop is called the lariat, and for you true Texans who are watching, you'll know that a lariat is a cowboy's lasso, so this structure got its name after the cowboy's lasso, okay, the lariat. And then you see what you do? You excise that intron. You excise the lariat, see? And that results in exon 1 being right next to exon 2. So again, what do you need to know? The splicing out of the introns, the splicing out or excision of the introns is accomplished by the spliceosome, which removes the intron through splicing, RNA splicing, and that results in exon information, remaining in the mature mRNA. Okay, so let me go back to the board and show you what alternative splicing is all about. But before I get to the board, let me tell you, alternative splicing is something that is unique to eukaryotes. Prokaryotes cannot do alternative splicing because prokaryotes don't do this splicing, do they? You know how the spliceosome splices out the introns? Well, eukaryotes take advantage of that in order to make more gene products than there are number of genes. So, for example, if I could splice out an exon, that would give me a different protein than if I left that exon in. I'm going to explain this at the board better for you, but just be aware that the human genome only has roughly 25,000 different genes. But the number of proteins we make is actually far larger than that. We have 80,000 different proteins because we have more than 80,000 different mRNAs. How is that possible? If I told you a gene's job is to code for a protein, how come there's 25,000 genes but more than 80,000 different mRNAs for 80,000 different proteins? How is that possible? Well, because of alternative splicing. Alternative splicing explains how eukaryotes can have more gene products, which means more proteins, than they have number of genes. Okay, so let me take you to the board and explain how alternative splicing accomplishes this.
All right, so here I'm going to explain alternative splicing. It's a process only found in eukaryotes, and it takes advantage of the fact that eukaryotes have exons, multiple exons. Like, remember this example I showed you earlier, exon 1, exon 2, exon 3 for this gene. Remember what I said? The entire gene is copied, so you get a pre-mRNA that's quite long. It includes all the exons and all the intron information, and then you splice out the introns to give you an exon. You splice out the introns to give you a mature mRNA that contains information for exon 1 next to exon 2, right next to exon 3. So all the exons are now right next to each other, ready for translation, right? So what's alternative splicing? Alternative splicing is, again, only found in eukaryotes. It is a process by which eukaryotes can make more than one protein, right? More than one gene product from the same gene. How can you possibly do that? Well, what if you shuffled the exons around or left an exon out or added an exon? Let me tell you. So for example, if the ribosome were to come along and translate this, right? Translation. If the ribosome were to translate this, it would translate exon 1, exon 2, and exon 3 information into a polypeptide protein, right? Into a protein. But what if I did an alternative to this splice, an alternate splice? So for example, what if I during splicing, you know how I spliced out my introns? What if I also, for example, spliced out exon 2? Did you see that? What if I also spliced out at exon 2? Would that not result in a much shorter mRNA with exon 1 right next to exon 3? Because we've left out exon 2 information. So now you have a really short mRNA that has different information. It's got exon 1 and exon 3. You've left out the exon 2 information. Now, when the ribosome makes that protein, is it going to make the exact same protein? No. This protein is going to be translated differently. It's going to be a shorter protein, and it's going to have a different structure to it, right? So in this case, the same gene gave you two different proteins due to alternative splicing.
All right, so moving on with the chapter, we've just finished our discussion about transcription, and that is the process of how mRNA is made. The gene information is copied to mRNA. Now we're going to turn our attention to translation, how the mRNA information is deciphered by the ribosome to produce the protein, the polypeptide. Okay, so what you need to understand, let me flip to this slide. I like to show it. Sorry, it's being a little laggy. Okay, what you need to understand is a simple concept before we get started. I want you to understand that the ribosome looks like this. Remember, the ribosome has a large subunit and a small subunit, each of which are made of part protein and part ribosomal RNA, also called rRNA. This was covered in Chapter 4, the structure of the ribosome. Again, you've got the large ribosomal subunit and the small subunit. I called it the large hamburger bun and the small hamburger bun. Now, what you need to understand is that this is the ribosome, the organelle responsible for translation, right, making the polypeptide protein. And how does it know what order to put those amino acids together, how to make those peptide bonds to make the protein? Well, what I need you to understand is that the ribosome is kind of like a 3D printer, okay? Think of the ribosome like a 3D printer. What does a printer do? It prints. And in the case of the ribosome, it prints proteins. So what you get at the end is the polypeptide chain. It's a bunch of amino acids linked together with peptide bonds. And the ribosome is the one that decides which amino acid to put next to which amino acid, and then produces it catalyzes the protein bond or the peptide bond between those amino acids. Okay, now how does it know how to link those amino acids? In what order? Well, it reads the mRNA. You see the mRNA? The mRNA is the information from the gene. It's the complementary copy of the template strand of the gene, right? So you have your mRNA, and here's the 5' end of your mRNA, which probably, if it says a eukaryote, it's got that cap, the methylguanine cap. And the ribosome will attach to somewhere near the 5' end of the mRNA. Now, what you need to understand, again, I said the ribosome's kind of like a printer. Okay, that's a good analogy for it, because printers are not useful without ink cartridges, are they? Or some kind of cartridge, right? Well, the ribosome really is like a printer. It uses the mRNA as information, but then it uses print cartridges that fit inside of these cavities. You see, these are cavities inside of if we had 3D glasses, we could see inside of the ribosome, there would be 3 cavities, the E site, the P site, and the A site. And that's where the printer cartridges go. Okay, that's where the printer cartridges go. Now, the printer cartridges are called tRNA. tRNA, which is basically, it's transfer RNA. tRNA is transfer RNA, These are short RNAs that form a clover shape that basically looks like this inverted L. Okay, this is a piece of RNA that looks like an inverted L. And these are the printer cartridges for the ribosome. So the ribosome will use these printer cartridges called tRNA in order to 3D print a printer. Okay, I'm sorry, in order to 3D print a protein. Okay, the ribosome is like a printer. These tRNA are like printer cartridges. Okay, and the protein is the product. The protein is the 3D printed result. Okay, so why are these short tRNAs important for translation? Well, by the way, there are thousands of these floating around in the cell. These tRNA, they're floating around. And what you need to understand is there's two important parts to this tRNA. This tip called the acceptor end, which is on the 3' end of the tRNA, and then this end, which forms a loop. This loop here called the anticodon loop, or the anticodon end. So this part of the tRNA is important, and this part of the tRNA is important. Why is this part important here, the acceptor? Well, because it is preloaded, just like when you go to the store and you get preloaded print cartridges, like pre-filled print cartridges. This end of the tRNA is preloaded with a amino acid, a very specific amino acid. And the enzyme responsible for that, you don't need to know this, but it's called aminoacyl tRNA synthase, or synthetase. This enzyme, what it does is it takes tRNAs and it adds the appropriate amino acid, in this case tryptophan. It adds that amino acid to the acceptor end of the tRNA. So look, now you have the tRNA with the appropriate amino acid attached. You see tryptophan is attached. And this is called a charged tRNA, which means the tRNA has an amino acid preloaded. And guess what? This tRNA is now ready to be used by the ribosome as a printer cartridge. Okay, so how is this tRNA used as a printer cartridge? Well, what does the ribosome have? The ribosome has these EPA sites, and the printer cartridges fit inside of these sites. Notice that these cavities look like inverted L's, much like a tRNA. The tRNA fits inside. So the way the ribosome knows which tRNA to use is that the tRNAs have anticodons for these codons. You see what I'm circling here? A codon. 3 letters, right? 3 letters, like AAG or CAG. These 3 letters. Well, the anticodons, you see here, the anticodons recognize those codons and attach to those codons. So the tRNA with the amino acid tryptophan, its anticodon would complement the, let me show you, tryptophan. Where's tryptophan? Here, right? Tryptophan. What's the codon for tryptophan? UGG. So UGG means tryptophan. So what's the anticodon to tryptophan? If the mRNA says UGG and it codes for tryptophan, what does the tRNA say in its anticodon loop? It does not say UGG. It has to complement UGG on the mRNA. So it actually says ACC. Does that make sense? So look at this. The tRNA for tryptophan, it has an anticodon. It will attach to the codon for tryptophan, and its anticodon would be the complement for that. Okay? So again, if the mRNA says UGG, the anticodon on the tRNA would say ACC, and that tRNA would be specifically preloaded, precharged with the amino acid tryptophan. Okay, so that's kind of how it works, and this is how translation gets initiated. How the ribosome begins translation. The small subunit, the small hamburger bun, right? the small subunit of the ribosome finds the mRNA, and it finds the 5' end of the mRNA. That's where translation begins. It also finds the start codon. It moves over until it finds the start codon. Do you remember what the start codon is? Look, the start codon is AUG. That's the start codon. And the start codon also codes for an amino acid, right? Methionine. So let's take a look. Look what happens. The ribosome finds the AUG start codon, and it also finds the tRNA that has the anticodon for that. So if AUG is the start codon, what is the anticodon for that? UAC. UAC. So the tRNA, only the tRNA that says UAC in its anticodon loop will bind, and it will bring an amino acid. You see this green dot is the pre-charged amino acid, the charged amino acid. And which amino acid must this green dot be? Look, it's methionine. It's methionine. Makes sense, right? Because the AUG start codon codes for methionine. So methionine has to attach to that codon. And why? Because the tRNA that carries methionine has the anticodon UAC. Okay, so hopefully that made sense. So once the small subunit of the ribosome has found the 5' end of the mRNA and also the start tRNA, which carries this green dot, which is methionine, then the large subunit, or the large hamburger bun, I call it, the large subunit is able to attach, and you've initiated translation. Okay, so from here, what happens? From here, the next tRNA is going to attach to the A site. You could think of the A site as the acceptor site. It will accept the next tRNA. Okay, so let's see that happen here. Look, you've got in the P site, let's say this middle one's the P site. In the P site, you would have AUG codon, so the anticodon would be UAC. It would be attached to this charged tRNA. What would the green dot be? Methionine. Well, then in the A site, that's going to accept the next tRNA. So the next tRNA attaches to the A site. That's this one with the blue dot. Okay. So if the DNA, if the mRNA said GGG, what would be the anticodon on this tRNA? If this said GGG, the tRNA would have to say CCC. And what amino acid must be attached? Look, what amino acid must be attached? Well, let's go back to our table. If it's GGG, that codon codes for glycine. Do you see that? GGG codes for glycine and only glycine, right? So, look at this. If these three letters, if this codon says GGG, the tRNA with the anticodon CCC would attach, and it would bring with it what amino acid? It would bring with it glycine, only glycine. Hi, Tig. Do you want up? Do you want to come say hi to your adoring fans? Tig's here. We can't have the final video without a cameo from our favorite cat, Tig. All right, so this is what happens. Again, tRNA comes along. tRNA attaches to the A site of the ribosome, and it brought with it the next appropriate amino acid. You see, the next amino acid as dictated by the mRNA, which is essentially the genetic message, right? So then what happens? Then something special happens. The P site does something important. The P site can make the peptide bonds, right? So look, what happens at the P site is this amino acid. Oh, you guys see Tig? He's being cute. Sorry. Here you go. Wonder if I could tilt this a little bit so you can actually see him. Okay. So here's what happens in the P site. This amino acid is cut is hydrolyzed from this tRNA, and a peptide bond is formed between this carboxyl group, right? Remember, amino acids have an amino group, a carboxyl group. There's a bond that forms between this carboxyl group and the amino group on the next amino acid. So now, as a result of that peptide bond formation, this tRNA is now called uncharged. You see, this tRNA no longer has an amino acid attached, right? And this amino acid has, this tRNA has 2 amino acids attached. Did you guys see that? So basically, you took the green ball and you attached it to the blue ball. And now you have the green ball attached with a peptide bond to the blue ball, and that's attached to the tRNA. Does that make sense? So you've made your first peptide bond. Hey guys, sorry about that. Tig decided to rest his face right on the F10 or F9 key, and one of those two cancels the video or turns off the video. So let me pick up where I left off. So again, the P site on the ribosome is able to, the P site on the ribosome is able to make a peptide bond and shift the amino acid from that tRNA to the amino acid on the next incoming tRNA, and produce the first peptide bond. So now you have 2 amino acids linked with a peptide bond, and you're on your way to making a protein. Okay, so at this point, what has to happen? So at this point, we've entered what's known as the elongation cycle. You've got a tRNA in the P site, a tRNA in the A site. Remember what happens? The amino acids from the tRNA in the P site shift to the tRNA in the A site. So now you have 3 amino acids on the tRNA in the A site, 0 amino acids on the tRNA in the P site. And then the ribosome actually moves to the right. The ribosome shifts to the right by 3 nucleotides, so 1 codon to the right. That means the tRNA in the P site shifts to the E site, the tRNA in the A site shifts to the P site, and the A site becomes empty, right? So you've got the tRNA from the P site now in the E site. You could think of the E site as the ejection site, where the tRNA gets ejected out of the ribosome. The tRNA with all the amino acids attached is now in the P site. And what's going to happen in the A site? That's right, the A site accepts the next tRNA. And this is called the elongation cycle. And every time you do this elongation cycle, every time you go through this cycle, then you have the amino acids extended by 1. So you have 1 more amino acid added on every step. There is a video here that is not my video, but it's linked here in the slides. I'll also put a card to it in the YouTube video right now. But this is actually a really good video showing you what's happening during translation, and especially during this elongation cycle. So again, new tRNA come in at the A site, uncharged or used up tRNA eject out of the E site. And what's the P site responsible for? That's right, peptidotransferase activity, making a peptide bond. I just used a long word. Let me tell you. Hold on, where is that? Yeah, do you see this? Peptidotransferase? Peptidotransferase activity is what the P site is up to. The P site makes the peptide bonds, right? That means it has peptidotransferase activity. So again, just for the exam reasons, what do you need to know? During initiation of translation, the small subunit of the ribosome finds the 5' end of the mRNA. Specifically, it finds the AUG start codon. It then grabs the correct tRNA, the one with the anticodon UAC, which complements AUG. It brings with it methionine, the start amino acid. You might want to know the start amino acid. It binds to the start codon AUG. And then the large subunit of the ribosome attaches. From there you have elongation, which means new tRNA come in at the A site, that match up with the codon in the A site. Then the P site does peptidotransferase activity, which means you shift all of these amino acids from this tRNA to the next amino acid on the incoming tRNA, which leaves this tRNA uncharged. And then once the ribosome moves forward, the uncharged tRNA, it gets ejected out of the E site. Just like when your printer is out of ink, you eject the ink cartridge, the spent ink cartridge. Fresh ink cartridge comes in at the A site. Uncharged tRNA exit out the E site. And the P site is responsible for what? The P site is responsible for peptide bond synthesis. Or the fancy word was peptidotransferase activity. So just know that for the exam. What does the A site do? What does the P site do? What does the E site do? And that's how you make a protein. That's how you make a protein. So you can see it again here. The fresh tRNA come in, the charged tRNA with the amino acids, they come in at the A site. The P site's responsible for peptidotransferase activity, making the peptide bonds that increase the length of the polypeptide by 1 amino acid. Then the ribosome moves to the right by 3 nucleotides. This uncharged tRNA moves to the E site. This tRNA with all the amino acids attached goes to the P site, leaving the A site empty. The next tRNA will come in at the A site. The uncharged tRNA is ejected out the E site, and the P site's responsible for the next peptidotransferase activity. And when does this end? How do you terminate translation? Where does it end? Well, eventually you have a really long chain of amino acids, right? And that's what a protein is. Do you guys remember a protein is just a really long chain of amino acids that folds into a tertiary structure, right? So at some point, you've made a really long chain of amino acids because you've done the elongation over and over again. And what do you find in the A site? If you look closely, the A site has one of the 3 stop codons. Do you remember the stop codons, UAA, UGA, or UAG? Those are all the stop codons. Any one of the 3 stop codons in the A site will recruit this guy. Here it's colored purple. It's called release factor. What you need to know about release factor is that it's not a real tRNA. It's actually a protein. And it recognizes the codon UAA. So it will stick to UAA or UGA or UAG, any of the 3 stop codons really. And once it does that, it lives up to its name. It's called release factor because it's going to promote dissociation and release of everything. The protein will float off and fold into a tertiary structure. The tRNAs are released, the release factor is released, and the large and small subunit of the ribosome also float off. All right, so again, this all has to do with the start codon and the codon table, the stop codons. These are the 3 stop codons I just talked about: UAA, UAG, UGA, and then the rest of these amino acids are coded for by different codons. Okay, so that's great. Now we've learned all about gene expression. There's only one more concept to learn about, and that's this concept of point mutations. So how do mutations work when you alter a single base? What you need to understand is that mutations are when you change the DNA. Like if I have the insulin gene and I change one of the I'm sorry. If I change one of the nitrogenous bases, if I change like an A to a G or a C to a T, that's called a point mutation, where I alter a single nitrogenous base in the DNA, in the gene. That could result in different things. So if I change one to another, it's called a base substitution. There are three outcomes of base substitution point mutations. One is called silent mutation, where even though the gene was mutated, it didn't affect the amino acid inserted. Another one's called a missense mutation, which actually results in the DNA being changed and a change in the amino acid inserted. And then there's a nonsense mutation, where the change causes a premature stop codon to form. So let me tell you an example of a silent mutation. Let's start with a silent mutation. So what if I had a gene and part of that gene had a codon CAA, right? The gene had a codon CAA. What if that gene got mutated to CAG? Okay, so CAA got mutated to CAG. How would that affect which amino acid is placed by the ribosome? Well, there would be no effect, right? So even though you mutated your gene from CAA to CAG, you didn't actually change the amino acid you use, and that's the definition of a silent mutation. You change the DNA, but you don't change the amino acid sequence. What about a missense mutation? By the way, you don't need to know the difference between transitions and transversions. So what about a missense mutation where you change the DNA and that changes the amino acids? So let's talk about that. What if I had CAA on the gene somewhere? That's a codon on the gene somewhere, and that got changed to CAC. CAA became CAC. You see how that's one substitution, base substitution, point mutation? Well, now the ribosome, instead of reading CAA and putting glutamine, now it reads CAC and it puts histidine. I'm getting my words wrong today with my amino acids, but CAA glutamine becomes CAC histidine. All right, so that's a change. You've substituted one letter in the gene, that actually changed the amino acid around. So think about it. What happens if you have the wrong amino acid in a protein? Well, the protein might not fold correctly, right? Remember, proteins have to fold correctly to function. And if you put the wrong amino acid in the wrong spot, that can actually cause the protein to not fold correctly. Does that make sense? Remember, because you have to, it's those R group interactions that have to form for the protein to fold correctly, right? So what else? Oh, by the way, before I move on, that's exactly what sickle cell anemia is. Remember, sickle cell anemia, you have this gene, and this is the gene for beta hemoglobin, right? CTGACT, right? This is the sequence for the hemoglobin protein. But sickle cell is an example where you've taken GAG, right? The gene has been mutated with a base substitution. GAG became GTG, you see? So an A became substituted for, or substituted by a T. So now now the codon reads GTG, which codes for valine, a hydrophobic amino acid, whereas before GAG in the normal or wild type gene codes for polar glutamine, okay, or glutamic acid. So what's the deal? Now you have a non-polar amino acid where you should have a polar amino acid. This causes the beta hemoglobin to not fold correctly, and that's actually responsible for the concatemerization that happens between those hemoglobin quaternary structures, and actually responsible for the phenotype, responsible for those exposed hydrophobic regions, responsible for sickle cell anemia. Okay, so you can see here that a simple missense mutation can actually have a really devastating effect on someone's health. Okay, so, and the last type of mutation is called a nonsense mutation. This is when a premature stop codon is formed. So let me tell you, for example, what if my gene should have said CAC or CAA? What if my gene says CAA and then somehow it got mutated to UAA, right? UAA, or on the DNA it would be TAA, but you get my point. CAA became UAA. Well, now guess what? Instead of the ribosome reading CAA, putting down glutamine and then moving on to build the rest of the protein, now the ribosome reads UAA and it thinks, oh, it's time to stop building a protein. And you can imagine how devastating this would be if this nonsense mutation occurred early on in the gene or in the middle of the gene. You wouldn't even make half of your protein. So that's an example of a nonsense mutation, where you get a premature stop codon, which is particularly devastating, right? And that's it. That's it. That's the last concept of the chapter. Tig's just being very comfortable today. I just wanted to thank all of you for joining me this semester. I really hope, and Tig really hopes, that you learned a lot, that you're ready to move on to Bio 2 and towards your bio degree. And we really hope that you've taken some of these lessons to heart. You understand how a cell works. Because guess what? Now that you know how a cell works, you know how life works, because life is based on cells, right? Now you know why genes are important, how the genes code for proteins, how proteins are made, how they function, how mutations result in altered protein function. Okay, TIG helped out a lot. How cancer works. We talked about how cancer works. We talked about, life in general. You understand all these cool concepts of pH and chemistry. And it's exciting. It's an exciting time for you guys because you're starting your careers as biology majors. And I really, really hope that you took some lessons. I'm going to be adding to this site. Tig and I will be here for years to come, hopefully adding more and more videos all the time to go into concepts in more detail. So, in the future, keep an eye out on the YouTube channel. Keep an eye out. Who knows? Sometimes you might want to refer back to these videos and see some new ones I put out to see new concepts explained. Keep in touch. Always keep in touch. And it's been super fun making these videos for you guys. Tig and I, it's a pleasure to do this for you. Always let me know. Leave any questions or comments. I'll try to respond. And thank you. And good luck to you. Good luck to you on your journey towards a biology degree and whatever you're trying to accomplish in the health or research professions. A very noble cause. And Tig and I wish you good luck. So with that, we'll catch you next time.